Hello, today we're going to learn about an experiment called ChIP Seq. As scientists, we are often interested with the question of what areas of the genome are actually occupied by my protein of interest. And we're forced to try and answer this question in the context of the cell, where things can get really messy. So, take a look at this visual of chromatin. There's so much going on. Proteins flying on and off, complexes forming, etc. Yet, there is a way to capture this experimentally. For example, this might be a transcription factor that regulates the activation of, for example, this target gene. To test this, we're going to use an experiment called chromatin immunoprecipitation sequencing. And there are several objectives to this experiment. Firstly, we will use peak calling to identify what chromatin my protein of interest is actually binding to. From there, we can characterize the motifs or do motif analysis on the called peaks to determine if there's significant sequence enrichment in the ChIP peaks for a particular sequence. This is helpful in the case of transcription factors, which actually do bind to a particular sequence. From there, we can annotate the closest genes and determine a list of target genes that might be regulated by the protein. And we can also determine what function of those target genes are, although that is less of the actual ChIP Seq analysis and more downstream. Now let's take a second and see how this experiment is done. The ChIP Seq experiment starts with cells, where our protein of interest is natively interacting with the chromatin. And the first step is to cross-link the protein to the chromatin by either UV or formaldehyde cross-linking techniques. After that, typically through sonication, the chromatin is fragmented into smaller pieces. From there, we are going to do an immunoprecipitation on the protein using an antibody specific to our protein. Then, we digest the protein, leaving the DNA bound to our protein. From there, we'll prep the library and send the samples for sequencing. And then the analysis begins. We will map our DNA to the genome without splicing. Let's see what happens now. The output of the analysis is that we can define a ChIP peak. So for example, you can see here we have defined a ChIP peak. There's enrichment. The way that we define a ChIP peak is that there's enrichment beyond the background. Additionally, from this, we can also generate motifs. And we can map those motifs to see where they are in the chromatin. Besides doing a ChIP experiment yourself, you can use ENCODE, which is a repository of ChIP Seq data, methods, and all things related. For ChIP Seq, they have numerous samples of many kinds of organisms and many cell lines where you can access data. Importantly, ENCODES actually sets the standards for ChIP Seq, and for an experiment to be published in the repository, it must meet ENCODE standards. Let's talk more about the ENCODE standards and other factors to consider in your experiment. Cross-linking. A key step that can make or break your ChIP is cross-linking. If you under-cross-link, you will lose peak signal. Typically, cross-linking occurs by adding formaldehyde directly to the media. And in this cross-linking reaction, new intra and intermolecular bonds are formed with other proteins and DNA. Interestingly, this step is time sensitive and temperature sensitive because this is actual chemistry going on. So if you over or under cross-link, you will notice the difference between your peaks when you call them later. The next step is fragmentation. You'll next need to optimize the sonication conditions so that the DNA fragments are the appropriate size. If the fragments are too small, then you'll have a challenging time mapping, and if the fragments are too large, you'll have low complexity. Lastly, your antibody. A ChIP-grade antibody that meets ENCODE standards is crucial. So the antibody needs to follow the criteria for what a ChIP-grade antibody looks like. And to do that, it needs to be able to bind natively. It needs to bind to the native protein. So, For every ChIP experiment, you will need an input control. There are two kinds of input controls. The input control serves as the background for the ChIP experiment. And so the first kind is what we're doing is we're cross-linking and fragmenting DNA, but we are not doing an immunoprecipitation. So this is a background for all the sequencing that we can do here. The second, which is the more stringent background, is a mock ChIP. And the mock ChIP uses the same procedure, however, it uses an antibody that reacts with an irrelevant antigen. Now, when calling peaks, the program compares the input to the experimental and finds a difference. So capturing background will help us distinguish the real peaks from the ones that should not be called. So if you see here on the right, you can take a look and notice that the ChIP peaks where there is a well-sequenced background, we are able to distinguish between what is background and not background. An important thing to note is that you need to have equal read depth on your input and your ChIP. Additionally, we are also going to need to talk about replicates. ENCODE necessitates 2 biological replicates, and any experiment that's published in the literature should have at least 2 biological replicates. Okay, now let's talk about actual peaks. There are several kinds of peaks. There are narrow peaks, which we can also call point sources, which are primarily transcription factors and have small ranges in the 50 to 100 pair base range. On the contrary, broad peaks from chromatin modifiers are large and can span across long ranges of DNA. So, identifying the type of peak is now going to be an important step in generating the library and performing the ChIP Seq analysis. So, one of the things to consider is whether you're going to be doing single or paired end sequencing. More commonly, you're seeing paired end sequencing being done now. But as we've talked about in the past, what the difference between single and paired end is, that will be important in your analysis. But there's going to be different library standards, and more importantly, for what type of peak you're expecting. So for example, if you have a transcription factor and you're expecting a narrow peak, you're going to want 10 to 50 million reads per replicate. So a total of at least 20 million reads between your 2 replicates. The higher the read depth that you actually have reveals low affinity peaks without saturation. However, as you can imagine, if you're on the higher end of your reads, you are going to saturate out the peaks that are really, really well defined. On the contrary, broad peaks, you're going to need some more read depth, at least 20 to 100 million reads per replicate. Now, let's talk about actually evaluating what you get off the sequencer and what the quality of your data is. You'll need to find certain metrics to determine the quality of your data, and I'm going to present some simple calculations here. The first is the non-redundant fraction. Okay, this is the number of unique start positions of uniquely mappable reads over the total uniquely mappable reads. All this is a measure of complexity. Is my data, am I getting reads that are all stacking on top of each other, which could be the product of over-amplification of a few actual regions? Or is my data actually reflecting this? Next, we're going to talk about FRIP. FRIP is really good for ChIP because we want to know what fraction of our peaks are actually in, or what fraction of our reads are actually in peaks. So you can look at the total number of reads and peaks and the total number of reads. And then you can just calculate a FRIP score. The expected FRIP score is 1 to 5%. Now, as you can see here, a higher FRIP score would give you a higher confidence in your peak. Additionally, there are lots of other metrics that you can use, such as the irreproducible discovery rate, the cross-correlation analysis, and the fast QC score that we have talked about a ton in this class. Although, what the best thing that you can do is that ENCODE at this website right here has a thorough list of standards that can be used. So with that, we are now going to go on to our next video. And yeah, that's it. Thank you very much.